Our hearing system senses sound pressure from in the order of 10 to the power 14. And that's a huge dynamic range. Looking at two different light-based techniques, and first one is called optical coherence tomography. We're trying to see if the light can help us look at a certain target chemical inside the inner ear.
Hey guys, it's Charles Beasley and welcome to Good Clinical, a podcast and YouTube channel dedicated to showcasing the future of medicine and human health. Today's episode is another in our series of our partnership with Te Titoki Mataora. Today's guest is Dr Haruna Suzuki-Kerr, who works in the field of sensorineural hearing loss. So welcome to the podcast, Haruna.
Great. Thank you so much for the opportunity.
Fantastic. Look, hearing loss is not something that most of us think about day to day, but I was quite surprised when we had our initial chat about this. It's quite a large problem affecting quite a large proportion of the population. I think you said 1 in 6 or 1 in 7 patients, 1 in 7 people throughout the country or globally suffer from some form of hearing loss.
Yeah, that's right. So it's a big problem. So statistically, 1 in 6 people are estimated to have some form of hearing loss. And almost a third of them are estimated to have what we call disabling hearing loss. So that's when hearing is bad enough that it can start to have an impact on your day-to-day communication. Globally, WHO's announced a report in 2021 that estimates about 1.5 billion people all over the world are affected by some form of hearing loss and expected to increase, obviously, with aging population. So it's projected to be as big as 2.5 billion people affected globally.
Wow. Okay. And there's different types of hearing loss within that. So hearing loss is a very wide bucket, shall we say. And sensorineural, which is the field that you're in, that's a specific type of hearing loss. And what are the others?
Yeah, that's right. So when we talk about hearing loss, we obviously think about ear, and we're all about ear. But depending on which part of the ear is affected to cause hearing loss, and there's a different type of hearing loss. So when you have, there's a part that we typically consider as the outer ear, and then there's a middle ear, which is a little space where when you're a child, you get infected all the time with middle ear infection, and there's a part that's called inner ear. And the inner ear is a part that's really deep inside your head where the sound is actually sensed and sent to the brain as an electrical signal. Now, for little kids, often hearing loss is called conductive hearing loss, where you have middle ear infection and your sound has a trouble getting into the inner ear. So that's one type of hearing loss. But the large majority of hearing loss cases are what we call sensorineural hearing loss. And that's when something bad happens in the inner ear, the most deepest part of our hearing system. And when things happen to this deep part called the inner ear, changes are usually irreversible. So once you get the sensorineural hearing loss, it becomes irreversible and you can't get your hearing back. And unfortunately, that's a large majority of hearing loss.
Right. Yeah, because whenever I think of hearing loss, a bit like what you mentioned, it's glue ear, having grommets put in because you've got the chronic infections, or you have a perforated eardrum, so that conductive aspect of hearing loss. But not much is really talked about that sensorineural, deep within the head, really complex architecture of the hearing to brain kind of apparatus. It's really quite amazing. And that's the majority of hearing loss because it's more common as you age. Is it just age-related decline that hearing loss happens, or are there other ways in which you can have sensorineural hearing loss?
Yeah, so there are many risk factors known already about sensorineural hearing loss. So aging is definitely one. With age, our hearing capacity changes. Yeah. Exposure to too loud noise for too much can also cause sensorineural hearing loss. And other things like certain drugs or certain medical condition can also be a risk factor for sensorineural hearing loss.
And what's happening when you develop that hearing loss? Is it just the tissues degrade with age-related hearing loss, or is it when you hear too loud of a stimulus for too long, is that damage to the conductive or the electrical apparatus?
Yeah, that's a great question. So our hearing inner ear is actually a very complex little structure.
Yeah.
So when we say inner ear, we are talking about organ for hearing called the cochlea. The term cochlea comes from a Greek term meaning like a shell or snail. And it's called so because our hearing organ cochlea does have a shape that looks like a little snail. So if you pick up a little snail seashell, that's how it looks like. The human cochlea is about 1 centimeter or less in size, and it's very small. It's a size that can sit on the tip of your finger, and it's deeply embedded inside your skull. So if you look at your ear and think maybe 3 or 4 centimeters deeper towards the center of your head, that's where it is. And in this little pea-sized snail shell, there are sensory cells that can sense sound waves and convert it into electrical signal. There are auditory neurons, or very, very specialized neurons for passing that sound information really fast to the brain, as well as the other cells that sort of make up the batteries or energy for these sensory cells and neuron cells to work. And we call it sensorineural hearing loss because changes can happen to either sensory cells or the neurons or the battery cells or a bit of both. And so it's very complex, and the difference can be, changes can be in any of these components.
Right. Is it so with like external stimuli? Is there more likely to damage one part of those 3 components? And with age-related decline, is it more likely one of the components to be damaged, or is it just a real mixed bag?
It's a real mixed bag, and it's an excellent question.
Yeah.
So we know from animal studies that, for example, loud sound can really be bad for sensory cells, but over time, neurons also get affected, and so are other components. And I think one of the challenges that we'll touch on soon is that we don't necessarily know what's exactly happening in human sensorineural hearing loss because it's so complex, and it's very hard to see what's happening in human.
Yeah, I mean, that's a good segue into studying this problem, because you can't. How would you study it? I mean, me, a layman for this topic, I would think, well, it's deep within the skull, 3 centimeters deep. You have multiple different components, all doing fairly different things, and you can't ethically, do a test whereby you compare a control versus, exposing it to bad stimuli. How do you study hearing loss in people?
Yeah, so that's really a Holy Grail question, Charles, that we're working on. So, so for example, if you experienced hearing loss, suddenly you wake up and you can't hear your child, which may not be a bad thing, but you have this problem with your hearing, and then you receive an audiogram to assess your hearing capability, and that's where you hear high-pitched sound and low-pitched sound of various loudness, and you report what you can hear. Now, this is a diagnostic test that assess what kind of frequency you can hear and how loud you can hear, but it doesn't say much about what's causing that change, the hearing loss. And as I mentioned, our hearing organ is quite small, so if you there's not much else you can do about getting more information about why you have that hearing loss. So you can look down the ear canal to see if it's ear canal or middle ear problem. But as for what's happening in the inner ear, there's not really any way for us to know. We can't easily take a biopsy from your inner ear because it is such a small thing inside your skull, and you certainly don't want open skull surgery just to see what's happening. Diagnostics like medical imaging, like MRI, the cochlea is a bit too small for that to give us enough information about if it's the sensory cells or neurons. So there's really a very much no way really for us to start to find out what's exactly causing your hearing loss that might be impacting you, right? In the patient right in front of you.
Gosh.
Yeah.
So that begs the question: if you don't know what the cause is of the problem, how can you create solutions for the problem?
Yeah. So that's So far, a lot of biomedical research in trying to develop, drugs for hearing loss is based on the animal studies, and that limits us to what we can model. So, for example, if you want to develop a drug for hearing loss caused by loud noise, you might have an animal model of noise-induced hearing loss by exposing animal to loud noise and see what changes happen and how we can stop it. Because we don't truly understand what's happening in human hearing loss, for example, there's a form called sudden hearing loss, or sudden sensorineural hearing loss, which affects people all of a sudden, as the name suggests, but we don't really know the cause. So we can't even have an animal model for that condition because we don't know what's happening in human. So, yeah.
Gosh, it's quite a tricky problem to solve because there's so many different potential causes, so many different components within the hearing apparatus that could be affected. And it's hard, or almost impossible, especially in man, to figure out which of those is damaged. So quite a complex problem that really requires a lot of thought and investigation.
That's right.
So how did you become interested in this topic?
Yeah, so I'm a biomedical scientist by training. Right. So I'm born in Japan, but I came to New Zealand and did my undergraduate and postgraduate degree doing biomedical scientist. And I'd have a little bit of research in Japan, and I came back here. And back then I was researching how vision impairment happens using animal models. But in 2018, I joined the hearing research team to start to understand where are we with biomedical science for hearing therapies and what needs to be done for some of these new drugs that's been developed in animal models to be translated to human clinical use. And then that was when I learned how hard it is to develop treatment for hearing loss. So, compared with eye, which might be a little bit easier to access, hearing loss, you really don't know what's happening in human. And that sort of made me really interested in, what can we do in the lab that can develop something that's really groundbreaking and make change in the way we treat hearing loss.
Yeah, so is the Holy Grail is to have a animal model that is really replicative of what is happening in human. So you can run different tests on, you can do different analyses, you can do, postmortems, I guess, to figure out what happened, this, that, and the next thing. So is that's the secret sauce to be able to take?
Good question. So what we are trying to tackle in terms of our Holy Grail is to develop a medical device, and this is what's been supported by the university and also networks and funders. So what we are trying to do at the moment is to come up with a novel medical tool, hopefully a handheld device in the future, which will go through the ear canal and get a little bit closer to the inner ear that I've talked about, and use light-based technology to get some information from the inner ear, a type of information that we currently can't get. Because that would help us understand what's happening in a patient in the ear and hopefully aid development of therapies to treat these hearing loss.
Yeah, okay. So it's answering that device would be developed to help answer the fundamental question of what's going on within the skull in those tiny little organs.
Yeah, that too. But, that understanding really bridges the gap because there has been a lot of research done using small animals to develop therapeutic compound, but there's a gap in that being applied to human clinical space. And that gap is actually inaccessibility and lack of understanding about humans. So if we can fill this space, we think it will accelerate the whole thing by bringing it together. Enabling more research and to be translated for clinical use.
Fantastic. So how did this idea come about? Was it a bit of a eureka moment, shall we say, for you and your colleagues in the lab going, okay, if we can get a device that can measure things closer, more accurately, so we can build an accurate picture, we'll be able to translate a lot of the previous research or future research into man. What was the genesis behind the idea for this future device?
So when I joined the BAC group in 2018, I was very fortunate to join a group of very experienced researchers at the University of Auckland, and also working with clinicians who have been helping this research project. And through the discussion, it became quite clear that inaccessibility of human cochlea was the major barrier in translation. And we sort of started to think about that ear canal device. So the projects, I'm involved with multiple projects at the moment. And the one I joined was to develop drug delivery strategies to deliver drugs to the inner ear. And that's a major impact of inaccessibility of the cochlea. And as we started to think about, how can we access the cochlea? And it also became very clear that, if we can't even administer the drug, we don't know how much drug got to the inner ear and which drugs to administer to the inner ear, also because of inaccessibility issue. So then we thought, that's actually really quite key piece of information too, that we need to know which drugs to administer to the right patient. So let's start working on that project. And that's what the second project that I'm working on with support from Te Titoki Mataora.
Excellent. That's super, super interesting. So this is early stages, right? So is the idea that the research that you're doing will inform the design of such a device?
So this project aims to, we are aiming to develop new technology based on light, and it's a collaborative effort. So when we thought, we want to find a way to get more information out of the inner ear, we formed a collaboration with physicists, because they are the expert in light-based technologies. They can make new light-based technologies. Our role was to find a model, then to test this, and we're using sheep as a large animal model. You might be surprised to know that the sheep cochlea is almost same size as ours, almost identical in size. Their ear canal in female use are also quite similar. It's like a kid-size ear canal. So for our ideas to develop a medical device that goes through the human ear canal and get the light-based information, sheep was going to be a very good model. And then clinicians that we've also grabbed clinicians to help us because they would be the ones using the medical device, and based on if our technology works, they'll be using it on the patient to then decide which drugs to administer. So their input was critical. So at the moment we're having a little bit of laboratory testing using sheep ear tissue as a model, taking it into a physics lab and to shine a different wavelength light and see what works. And then report that back to clinicians to see what they think. So we can start to build a picture and vision towards a prototype.
That's so interesting. Okay, we're going to have to unpack this for me because I'm a bit slow. But so... Why light exactly? Why is it the different wavelengths? As opposed to some kind of other tool for looking at what's going on in the inner ear.
Yeah, that's a great question. So the inner ear, the cochlea that I mentioned, lives inside the skull, and it's very small and. If you think about it, inaccessibility is because of the bone. Now, it turns out that in the inner ear, there is two openings to this bone. First is called oval window, and second is called the round window. Oval window is where you might have heard of a little ossicles or little middle ear bones. So that's latched onto the oval window to pass the vibration coming from the sound to the inner ear. And then the other window is called the round window. So the idea is that oval window is pushed by sound and round window bounces on the other direction. Now, this round window is naturally existing opening on the bone. So every other part is covered by bone, but this round and oval window. And the round window is covered by a membrane. And that membrane we know is translucent. So it's a naturally existing opening that we might be able to use. And what we want to use it for is non-invasively. So without poking through this membrane, we want to be able to just sort of target this window, sit in front of it, and without disturbing the sensitive machinery inside the inner ear to collect the information. And, light is non-invasive. It doesn't cause any harm as long as it's not too bright or, in any way. And we have our scientific hypothesis is that this window and a translucency allows us to use the light to non-invasively get some information from the inner ear.
Incredible.
And it's fast, light is safe, it's fast, and depending on the technique you use, it does let you visualize things and collect data.
Yeah. So what data is being collected? Is it of the three different components of the cochlea? So like the battery, the sensory, and the neuronal component of it? Or is it just looking at the health of the round window?
That's a great question. So first is the structure. So we are looking at two different light-based techniques. And first one is called optical coherence tomography. That's one technique that's used quite commonly to diagnose disease of the eye.
Yes.
And we are thinking of sort of building on that. Like using a similar approach, but come up with different ways to use it to get some information around the structure of the inner ear close to this round window.
Yep.
And the second thing we are doing is we are looking at another technique actually to see if we can detect some signature about the chemistry environment inside the inner ear. So the idea is to essentially shine the light through and collect what comes out. And depending on the light signature of the light that comes out, can that tell us about concentration of drug we administer in the ear? Or can we tell something about other compounds that might be in the inner ear side. So we're trying to see if the light can help us detect a certain target chemical inside the inner ear.
That's incredible. Okay. So it's about seeing what components of the inner ear might have been damaged by various means, whether it's age-related decline or, loud bangs repeatedly because your colleagues upstairs are really loud, for example. Or to be able to see if, say, a drug is getting to where it needs to go.
Yeah, that's right.
Wow, okay. All from using light. That's incredible.
Well, we're starting quite small. So at the moment, we're really focusing on how much can we get out through this window non-invasively, because that's what we are trying to achieve. We don't want to be invasive, and that means our target is a window. But we are sort of testing the concept at the moment.
Yeah. That's such a cool. But I imagine so. You've got to emit the light through the eardrum. Does it have to go through both windows, or is it just the round window?
So the idea is to go through the eardrum.
Yeah.
And so we'll go through the ear canal. We'll have to go through the eardrum through a little slit.
Okay.
And then stop right before the round window. So that would be the borderline between the middle ear space and the inner ear. So the probe will sit in the middle ear, aiming at the round window to see if it can collect the information from the inner ear. But it's non-invasive to inner ear that we haven't really gone into the inner ear.
Yeah. Okay. So just a small little cut in the eardrum, so you can get right up, right close to that window,
That's right.
and then shine a light into it, see what comes back, or see optical coherence tomography. Yeah. See the structure, find out what's damaged, and then treat accordingly. So you've actually got hard data on what is happening to that patient, as opposed to theoretical data.
Yeah, it's a little bit like that. So we're hoping that the combination of knowing what kind of chemical is beyond the inner ear and getting a little bit of understanding around this structure combined together might give us something useful.
That's incredible. Okay, so I imagine this is quite groundbreaking stuff.
We think it's got a lot of opportunities. To sort of fill in the knowledge. So because there's currently no way. So, for example, if we can detect what chemical is inside the inner ear through this round window membrane, there's no other way currently to get that information other than going in with a surgical procedure to sample directly from the inner ear. Now, that's highly invasive, and it does obviously require surgery. So considering there's no other way to currently get that information, we think if we can do it, it's pretty groundbreaking. But it's challenging. We have to acknowledge.
Yeah, absolutely. So would the idea be if the technology works and a device is feasible to manufacture and give to clinicians, would that be used in that same way? So there would need to be a small incision made in the eardrum to collect that data and then have a personalized diagnosis as to the cause of the sensorineural hearing loss for that patient, and thus the clinician could tailor-make a therapeutic regimen for them.
Yeah, that's right. And that's where we are quite fortunate to have clinicians on board and supporting this project. So, obviously going through the eardrum is semi-invasive. It's not non-invasive, and does require certain justification and come with some small risk. It is so currently there's a therapy called intratympanic injection, and that's when you have a sudden hearing loss. Clinician will go through the eardrum to inject steroid in your middle ear. And also earlier we talked about middle ear infection and grommets or cutting a slit. So eardrum does heal over, and with enough medical reasons, there's a justification to go through the eardrum. For our project, the goal is to get that information from the inner ear. We currently can't. So hopefully that can then help the patient to be treated with the right drug or as a part of to see if the drug actually gotten into the middle ear. So there would be a benefit that needs to be large enough for the patient's benefit to justify approach of going into the middle ear. And that's something that we are having a lot of discussion at the moment with clinicians, but also people developing drugs. Because obviously our medical device, needs to be part of that treatment regime with clinicians and drug and our device.
Absolutely. Gosh, honestly, that's kind of embarrassing to say, but I'd never thought very hard, never put much thought into hearing loss, but it is a very, very complex problem. And when you said how many people it affects, it's going to be, and it will be growing into the future with an aging population. And there's so many unknowns as well because of how difficult it is to access. It's a very interesting, must be a very intellectually stimulating pursuit.
Yeah, absolutely. Yeah. And it's an amazing system that I think we take for granted.
Absolutely.
So in terms of magnitude, our hearing system senses sound pressure from in the order of 10 to the power of 14. And that's a huge dynamic range. And we also hear, super high pitch sound to very low pitch sound. So it's amazing bionic machine. And very challenging to study and do anything, but it's a very exciting field with a lot of potential to improve people's lives.
Absolutely. So how let's talk about the potential commercialization aspect of this, because part of this whole podcast is how do we get great ideas like this one into a patient? How do we take it from the benchtop to a patient? How do we take it from the lab into the wider world? Because that's where, it's very intellectually stimulating. It's great for research and development, but the end goal should always be, how is this going to improve people's lives?
Yep.
And we can't, if it stays in the lab, it ain't going to get to a patient. So because you're working with clinicians and all these other stakeholders, you're obviously thinking about the commercial product this early in the development, which is great. And that's been supported by Te Titoki Mataora.
Yeah.
So why don't we talk about the support that they've provided, maybe to really put a more, shall we say, commercial kind of spin on this whole project.
Yeah, absolutely. I mean, so for us, the commercialization and innovation is a pathway of taking our lab research to the actual use by clinicians and for the people's benefit, like you mentioned. And we are very early on, but it's also something that we had to, and I had to learn quite a lot, how that pathway looks like. Like I mentioned, a lot of biomedical scientists like us, we all have a good goals, but we are trained in the lab to start with, and we needed to think about how to think about commercialization and innovation space. So Te Titoki Mataora, first of all, gives like a startup funding for ideas to discuss about the commercial pathway. So I've received seeding fund from Te Titoki Mataora, and that helped us, first of all, to have lots of meetings with clinicians and us researchers and new physics collaborators and also community members. So we are linked to iwi organization called Iwi United Engaged to hear communities' view as well and what's needed in New Zealand context. So it let us have a lot of conversation to plan it together. About how it should look like and what we can do to come up with a better plan. It also helped us to run a bit of a commercial report. So market research was done by someone with more of like a marketing expertise to look at what's the actual market's going to look like.
Yes.
Because when it comes to commercialization, it's actually about commercially viable solution.
Absolutely.
Rather than just a pure goodwill. And it is true, medical system is always under pressure too. So that sort of expertise and network and support, but also market research was done using this funding. And then the research funding from Te Titoki Mataora is also supported us to do some seed research experiment. So with that experiment, we had light-based techniques tested on the sheep round window membrane to collect some pilot data. And that's really important because we want to have some confidence before moving further that this idea is going to work and feasible. So some lab work, but as well as for us to develop the plan together, find new people that we might, whose expertise we would really like to have, and even to sort of have a conversation with the local community.
Yeah, that's fantastic. So, I mean, without, I've often said, a few guests have actually said to me that you need to very early on be thinking of the commercial implications of the research and using feedback from various different stakeholders, whether they be current or future, to design your experiments. Because if you don't have that input early on, you could start meandering down a road that has commercially like a commercial dead end, shall we say. Like a lot of input can be given early on to provide you with that context and that market research and that viability to inform, the early research that could then go into preclinical and then clinical models and so on.
Yeah, absolutely. And we are learning so much, not just from clinicians, but more recently, starting having conversation with pharmaceutical companies.
Wow.
And they are all overseas.
Yeah.
The company is developing new drugs for hearing loss based in Europe or America, different time zone. How do we pitch our case, and how do we have that almost like commercial conversation? All of that we sort of had to learn along the way. So having like network support has been really, really helpful, and their feedback has actually changed. How we plan our next step quite a lot.
Yeah. So if Te Titoki Mataora wasn't around, what would the project look like today? Would it be around, or would it be around in its current form or in a different?
Oh, absolutely different.
Yeah.
And I think, I think it would be still quite a lot heavily focused on just lab by itself, because what Te Titoki Mataora has really been great at is really encouraging us to have that commercial conversation early on, diversify the type of people we are talking to, and giving us some resource to go and meet people. So without it, we'll still be talking among ourselves, just between that inner ear biomedical scientist alone in a silo. So the impact has been huge.
Yeah, that's fantastic.
And hugely helpful in the right direction, I believe.
Yeah, I mean, these kind of things make me really excited because I think there's so much talent within New Zealand. We've got so many great science graduates. I did a biomedical science degree. You've done a huge amount of biomedical science research. But what's really going to take New Zealand forward and into the future is not just benchtop blue skies, research, but it's applying that research in the real world, commercial value, and getting people to, because not everyone can be a researcher, right? There's limited roles. A lot more people, if you build a pyramid really, the base of a pyramid really widely, you can build it really tall. So if there's more places for people to go, then there's more people that can lend their skills, and we're going to grow the economy and our living standards and blah, blah, blah. The whole thing. So I think stories like yours and others that Te Titoki Mataora, I always forget the first two, are a part of, I think it paints such a great picture for the future of translating biomedical research into medtech or health tech applications. It's really cool.
Yeah, so, I think so too.
What's the next steps for this project? So it's currently testing it with sheep inner ear components. So it's not live sheep, or is it on live sheep as well?
Not live sheep yet. And we are doing a lot of sort of sheep tissue at the moment, to get more clarity on what we can do with the technology, and also I think continuing conversation, because last few years supported by Te Titoki Mataora, we've learned huge amount about what pharmaceutical companies' perspectives are, and what are some of the real clinical constraints are. It's almost like you find out something and you feel like you know it, you ask someone else another question and you discover more things that you have to find. So I think we're making huge progress forward, but I think the immediate next step is just keep on going, accumulate a little bit more proof of concept data, but also expand our network and continue to have these conversations so we know exactly where we should be heading, and hopefully come up with big plan towards a live testing and prototyping.
Right. So Nick, and at that point that will be in a sheep model with a prototype, and then eventually you would be in a position where you could move into a human.
Yeah, I think that might be some years away still, but yes, absolutely. And sheep, large animal study testing is becoming quite a big thing. So a lot of drug development might happen in cell-based model or small animals, but when it comes to administering in human, including the actual procedures, the large animal model is becoming quite an important pathway for all the field, including inner ear therapies. And in New Zealand, sheep model is actually quite broadly becoming more popular in a lot of different fields like brain or other systems. So it's definitely a strength in the biomedical science in New Zealand that we'd also like to build as our strengths to compete on the global stage.
Yeah. I mean, there's 5 sheep for every human, right? So you don't have you're not in short supply of potential subjects. So the sheep model is so well suited because they have, you mentioned like a childlike dimensions of their ear canal and a similar size dimension of their inner ear. Obviously you couldn't do, like typically, pharmaceutical or biotech research is done on a mouse or a rat model, but I can't imagine that would be suitable for this endeavor. Were any other animal models considered for this, or is it just makes sense on the basis of those two?
So on the global stage, large animal space is either sheep pig or monkey. Now, monkeys in primate obviously comes a lot more ethical consideration, and I think it's only feasible in certain countries like USA.
Right.
For us, we considered pig and goat and sheep, and for us, because of what we're trying to do, the ear canal and overall geometry was quite important, and that was best for sheep compared to with pig, which has quite triangular head. So pig is great too in terms of the cochlea size, but their ear canal geometry is quite different.
Is that right? Yeah. Can't say I've ever given a thought to ear canal geometry before, but I can imagine.
I haven't either. Not before 2018.
There you go. Okay, so you'll do a series of testing in the sheep tissue, then move into a sheep model, further optimize the device, and then you'll have something that you can take into the clinic per se. But
that's
obviously,
the idea.
These kind of things they don't happen overnight. They don't. You can't conjure them up, in a week. They do take time, and they do take a lot of money as well. So I bet as part of this whole commercialization thought process, I imagine you're already thinking about where's the money going to come from to fund all of these endeavors, right?
Yeah, that's right. That's something that we spend a lot of time on, trying to come up with funding strategies.
Yeah, it's tricky.
Yeah, it's definitely a challenge. I mean, I think biomedical science in particular takes a long time, and so that's why I'm currently involved with maybe this is one of 3 projects that I'm involved in, all separately funded by different funding agencies. So my other project looking at blood flow in the cochlea is funded by Auckland Medical Research Foundation. This project, and it sort of links in with our wanting to understand what's happening in the ear. So this technology was really supported by Te Titoki Mataora. And the other project that I'm part of, run by my senior academic professor, Peter Thor, and that's supported by a Ministry of Business and Innovation. An entrepreneurship program. So it's a hard work.
Yeah. But I mean, you get a certain amount of money to kind of take it to the next step, and then you've got a different product or proposal to present to what might be the next layer or the next level of potential funders, whether it be additional seed or something else if it's an entity. That's really exciting, really interesting. Okay, so what would be your ultimate goal for this technology, this project? If you could have supreme aspirations for it, what would it look like?
Well, our big dream and supreme vision is this amazing handheld device which is used widely in walking specialized clinic. Patient can come in, we can put it into the ear canal, know what the patient's inner ear looks like. We'll admit, and based on that information, they'll receive a drug injection into the middle ear. They get treated. Then they can come back in a few weeks' time to have another assessment to see how they're going, to decide on a bit and the next treatment regime, and hopefully together that's going to stop this patient's hearing from getting worse, or even possibly recover, so they don't have to get deaf or think about getting cochlear implant surgery. So that would be huge change to the way we can treat hearing loss, and that would be my dream. Absolutely.
And it's a big market. A lot of people, like you said, how many billion by 2030?
2.5 billion. But that's some form of hearing loss, and it is quite a mixture. Yeah,
remarkable. Well, Haruna, thank you so much for your time. It's been really, really interesting. I'm really excited to see where this goes, and it's great that there's so much support there, particularly from Te Titoki Mataora. It's really exciting. If people want to find out more about your research or yourself, where can they go?
Yeah, so, we are based in the University of Auckland, and, we are always all part of Te Titoki Mataora too, so hopefully they can search up about us, and with Auckland University and Te Titoki Mataora websites and contact us. And we'd love to hear from people too. And we are quite proud to be doing this in New Zealand. So we're interested to hear from people who are interested in our work.
Awesome. Well, thank you so much.
You're welcome.
Cheers.
It's a pleasure to be here. Thank you.
Thank you.